Hello everyone. In this video, I am going to teach you the process of digestion. So first of all, how many steps are there in this process? I am going to tell you. The steps in this process is that ingestion. Ingestion means when we take food inside our mouth. Followed by digestion. Digestion is to convert complex food into the simple one. We'll take this point further. Next is absorption. That means the food which is digested, in what way it will be digested inside, it will be absorbed inside the blood. Next is assimilation, means the food which is absorbed into the blood or into the lacteal, it must be again reached up to the cells. Next, egestion, the rest food, undigested food, ultimately converted into the fecal, collected into the rectum, and then it comes out through the anus, that is known as egestion. So the main agenda is the digestion. The question arises, what is the need? Why we need digestion? The question is that what is there in our food? In our food, the components are carbohydrates, proteins and fat. What are the sources of the carbohydrates in our food or in our daily diet? That is your breakfast, your lunch, your dinner. So your things are, for the carbohydrates, in your daily routine, in your diet, you are taking rice and chapati, bread, pizzas, burgers. They are rich in carbohydrates. In what way we are taking carbohydrate? That is polysaccharide and termed as starch. This is starch. Now, starch is the complex food. Starch will be converted into the simplest form, that is to be termed as the monosaccharide. But just like to convert the complex one into the simple one, we convert this complex starch into the monosaccharide. There are simple intermediate steps also. So before converting into the monosaccharide, there is a middle stage that is termed as the disaccharide. Disaccharides means two molecules of the monosaccharides are here. That's why we are using the term disaccharide. Next, the component which is there into the food, that is to be termed as the protein. This protein is a building block. It is there for the repairment, and of course the protein is, its unit is amino acid. So ultimately with the help of enzymes, we convert this protein into the amino acid. But again, the intermediate step is there in between these two. And first of all, this protein, it's a complex food, will be converted to the peptones and proteoses, then amino acid. So it is written as amino acid is the unit. That means the end product of the protein digestion is the amino acid. Last one, that is termed as the fat. This fat will be converted to the fatty acid and glycerol. These are the two molecules which are actually digested with one molecule of fat. One molecule of fat after digestion gives you 3 molecules of fatty acid and 1 molecule of glycerol. So these are the 3 major components which are actually present in our food. There are certain more things like vitamins and minerals, but these are not there. Vitamins and minerals, that means minerals are minerals, like iodine, like iron, like calcium, magnesium, phosphate. But these two things are not to be digested. Essential part of the food because they regulate our metabolism. So this is your food. So what is the definition? The definition of the digestion is to convert the complex food. What are the complex food? Carbohydrate, protein, fat into their simpler units. What are those units? For carbohydrates, it is monosaccharide. I am going to underline this one. For protein, it is amino acid, already underlined. And for fat, it is fatty acid and glycerol. Now for this digestion, that is the conversion of complex into the simple one. For this digestion, we have two different steps. One is mechanical, also termed as the physical. In certain books, it is written as the mechanical. What does it mean? The meaning is, in this process, we are not using any type of the enzyme. Mechanical is chewing of food with the help of teeth, mixing of saliva with the help of tongue, churning of food into the stomach, that is known as mastication. So these are those things which are to be termed as mechanical one, and there is no need of enzyme. What is the next? That is the chemical. The chemical way is that using of the enzymes for converting this complex one into the simple one. So first of all, we'll start from this mechanical and chemical in the process of digestion, which will start from our mouth. Now, after the ingestion, when the food is there into the oral cavity and buccal cavity, so what is it? The first of all, the food is there, in the first position of the food is there into the buccal cavity. So here I have written as the buccal cavity. This is also termed as the oral cavity. So oral cavity is written over here, oral cavity. And in common mode, we normally use the term mouth. So inside the mouth, we have teeth and of course tongue. This tongue is attached with the muscles on the oral cavity, and those specific muscles are to be termed as frenula. So the tongue is attached into the oral cavity from the back side, that is termed as frenula. We have already discussed 3 pair of the salivary glands into the mouth, and that salivary gland is responsible for, those salivary glands are responsible for secreting saliva. Saliva contains salivary amylase, but along with it, mastication or the chewing of food is there. Food chewing along with the mixing of saliva with the help of teeth, ultimately the whole soul food, which is actually having chapatis and your cooked vegetables, maybe the pulses, ultimately turns into round ball-like structure, and that structure is to be termed as bolus. Then, in this saliva, which contains the salivary amylase, it is responsible for the digestion of starch. Starch itself is a type of, it is actually a polysaccharide, and this salivary amylase, the same one, the same salivary amylase is also termed as ptyalin. This P is silent, Tyalin. P is also there, but it is the silent one. So the starch, first of all, as we have already discussed right now, the starch will be converted to the disaccharide. So it will convert it into maltose. So this maltose is disaccharide. This is disaccharide. Disaccharide. Sometimes in your examination, a reasoning based question is to be asked. When we chew chapatis for more duration, for longer duration, in that condition, it turns into sweet taste. We know that the chapatis contain starch. When we chew, excess chewing is there, then this starch, which is not, it is not sweet in the taste, but due to excess chewing, the starch converts itself into the maltose. And maltose is sweet in taste, that's why the excess chewing of the chapati gives you a sweet taste in your mouth. Now, in this, along with the ptyalin, which is there into the salivary gland, the mucus is also to be secreted over here. This mucus makes the bolus slippery, and ultimately it slips down. And while moving downward after the mouth, there will be one more passage and one more digestive organ. That is not the digestive but the place, that is pharynx. That means after the mouth, that is bolus. Bolus will enter into the pharynx, and from pharynx it will reach into the esophagus. Esophagus. G-O-U-S. But in esophagus, a wave-like constriction motion is there. Due to this motion, due to this movement of the muscles of esophagus, food which is actually rich in mucus also, surrounded with the mucus also, it slips down, termed as peristaltic movement. So what is the path? The path is first of all buccal cavity, followed by pharynx, and then followed by esophagus. So these are these things. But remember this point, that in these two places there is no enzymatic action at all. Peristaltic movement itself is a type of mechanical function. Then food reaches into the stomach. The stomach is a pouch-like structure. Muscular wall of the stomach is again made up of involuntary tissues, involuntary muscles, and the stomach secretes gastric juice. When we use the term juice, that means it is a mixture of so many things. So what is gastric juice? Gastric juice includes HCl. This HCl is an acid, we know. The HCl maintains a very low pH in your stomach. This is approx 1.2. pH is actually the hydrogen ion, the logarithm value. So this is 1.2, the HCl. pH of this gastric juice is 1.2. Over here when we were there into the mouth, it is hardly 6.8. So this pH 1.2 is responsible for converting certain inactive enzymes into the active one. Now, here in the stomach, the food stays for at least for 4 hours. Now, the duration is going to increase. In mouth, we cannot hold or we cannot keep the food after making the bolus for a longer duration, and then there is no stoppage at all in the pharynx and of course into the esophagus. But when the food reaches into the stomach, then it stays over here at least for 4 hours. Here, this HCl is responsible for converting certain inactive enzymes into the active one. Inactive pepsinogen will be converted to the active pepsin with the help of HCl. Now, what is the function of this pepsinogen? Going to write down over here. That is, first of all, it is responsible for digestion of protein, and when it is active state, then it is to be termed as pepsin. And as we know that protein is the complex one, the first step is to make peptones plus proteoses. So this is the protein which is there into the food. It is actually there into the bolus, and when it reaches into the stomach, that means after 1 and 2nd and 3rd and 4th, ultimately after the complete meal, when we finish this one and the whole soul food is to be stored into the stomach, in this way the process is to be running. Next, the protein is the target of this pepsin is the protein, and the second inactive enzyme is to be termed as prorenin. This prorenin is also converted into renin because of the presence of HCl. Then what is the function of renin? Actually, this renin is responsible for digestion of the protein of the milk termed as the casein. We can say that after the birth and up to the age of approx 14 years, its function is there into the majority, and later on this enzyme is actually the volume is to be declined. What is its function? That milk protein is to be termed as casein. C-A-S-E-I-N. In bracket I am going to write down it is milk protein. This is milk protein. And you can observe that fat and major carbohydrate digestion is not over here. Even we started with the digestion of the carbohydrate. But in case of the stomach, the digestion of the protein is there. So this casein is the milk protein. Going to write down the name of enzyme, that is termed as renin, and this casein is converted into para-caseinate. This is termed as para-caseinate. So, digestion of milk protein, digestion of food protein, what we have eaten in form of pulses, and of course maybe in form of sprouts, maybe the egg in form of egg, that is albumin. So that protein is converted into the peptones and proteoses with the help of enzyme, that is pepsin. Stomach itself, along with this all enzymatic activities, the mastication of the food is there, churning of the food is there, mixing of the enzymes and action of the enzyme is also there. But ultimately it is converted into a pulpy type of fleshy, pulpy, slippery, fluid-like structure, and that structure is to be termed as chyme. First of all, we started from bolus, it was there into the buccal cavity, and secondly, after the complete digestion into the stomach, the physical state of the food is to be termed as chyme. This chyme means that the food is there in a form of pulpy, fluid-like structure. After stomach, that means after 4 hours, this chyme, last stage is the chyme, where digestion of the protein is completely done. That means the intermediate stages are here. Digestion of the milk protein is also completed over here, but you can observe the fat is still remaining. There is no digestion of fat at all. So the next is that ultimately the food reaches into the small intestine. As in my previous video, I have already taught you the digestive system. For your convenience, I'll post the link of that video also in the description box. So now the next step, that is the next organ, the digestive organ which is there after the stomach, that is termed as the small intestine. This is small intestine, having three parts. The first part is part number 1 of this small intestine. It is termed as duodenum. Right? Now what is the duodenum and what is its function? In what way the digestion will be here? This is the stomach. For your easy reference, I am making a very short diagram. This part is to be termed as stomach. This is stomach, and we know that the pancreas, we can say the pancreas is here. This is a gland. It is pancreas, and the liver part is here like this one. This liver contains gallbladder. Gallbladder is the storage device outside the liver, but the liver makes bile, and that bile is actually stored by the gallbladder, and a duct opens into the pancreatic duct, and the common duct which opens into duodenum, it is to be termed as hepato. Hepato, that means liver. Pancreatic, Pancreatic, means this one, pancreatic. That means duct from the pancreas also, duct from the liver also, ultimately unites to make hepatopancreatic duct. And from here, that means from this duct, a fluid is to be poured into the duodenum. This is duodenum. The food is here now. The part which is actually there in the form of the sphincter, it is to be termed as pylorus. From stomach, the food which is converted into the chyme will move from the sphincter and ultimately will reach into this duodenum. Here the mixing of pancreatic juice and the bile juice is there. First of all, going to tell you what is the function of bile. Bile does not contain any enzyme, no enzyme at all. But without this bile juice, which is yellow in color because of bilirubin and biliverdin, in absence of this bile juice, no digestion of fat is possible at all. What is the main function of this bile juice? The bile juice, earlier, that means in the stage where the food was there into the stomach, the pH was 1.2 approx, that is highly acidic. This bile is actually alkaline in nature. It is alkaline in nature, right? Because of the presence of sodium bicarbonate. Going to write down the salt of bile, that is NaHCO3, sodium bicarbonate. This bile converts a big lump of fat. Consider we have a big lump of fat like this one. This is a fat. To reduce the surface tension, that means in between, breaking of this fat into this, ultimately a big lump of fat will be converted into small droplets and termed as bicille. And it is to be termed as, this process termed as emulsification. Here I am going to write down an arrow. It is termed as emulsification of fat. Without this emulsification, digestion of fat is not possible at all. Not at all. And when it is converted to small droplets or a small micelle, in that condition the fat is to be digested, and then the fat will be converted into fatty acid and glycerol. This is fatty acid plus glycerol. Now the question is, the enzyme is coming from which gland? And the answer is pancreas. So this is pancreatic juice which is responsible for secreting the enzyme. Bile does not contain any enzyme. This is the pancreatic juice which contains enzyme. So the first enzyme is lipase, which is responsible for digestion of fat into fatty acid and glycerol. This is the correct one, that is termed as the fatty acid and glycerol, and this is emulsification of fat. Next is this pancreas also secrete digestive enzyme. This is actually responsible for the digestion of protein. So what is the name of that enzyme? One part of digestion into this duodenum. Here we are. This is the organ. Second enzyme is trypsinogen. This trypsinogen is again inactive. That means this trypsinogen will be converted to the active trypsin. What is the procedure? This trypsinogen is converted into the trypsin. I'm going to write down the name of the enzyme. It is trypsin, a protein digestive enzyme. But this process is also enzymatic. That means trypsinogen will be converted into the trypsin with the help of another enzyme, and that the name of that enzyme is enterokinase. Meaning of kinase is to make something new always. This is very much important, and of course the question of your exam, name the enzyme which is responsible for converting inactive trypsinogen into trypsin. Now as it is a protein digestive enzyme, so the function will be there on peptones and proteases. So this is peptones plus proteases, right? Going to remove this trypsinogen or enterokinase. Now the trypsin is ready with us. So this is again going to write down, this is trypsin, right? And now the dipeptides, in certain book it is written as polypeptides. In certain books it is written as polypeptides also, but the correct word is dipeptides. This is the correct. That means not the all. It's not the duodenum. It's not possible the whole complete digestion of protein will occur over here into the duodenum. It's a time-taking process. That means the peptones and proteases actually convert into dipeptides, but no doubt that the little bit amount of the protein is remaining into your food. Then the second part of the small intestine is to be termed as the jejunum. Again there is a simple movement of the food at all. No digestion, no digestive enzymes, and the third part is termed as ileum, and I am going to draw that here only so that you can identify that from where to where we all are going downwards. I'm going to remove this one. First of all, find out the location that when this is the large one, large intestine, like this, and in between this one, there is the ileum part, which is to be termed as the last part of the small intestine. Going to open this one like this. So now we are here termed as ILEU and ileum. So now the complete digestion of the food is there into the ileum. What is it and in what way we are going to digest? Now it contains almost all the enzymes for the complete digestion of the intermediate component which are actually formed into the duodenum also. So one by one, as we know that the carbohydrate ultimately reaches up to the dipeptides. That part is actually left, right? That is in pancreas. The pancreas, this CREA, this pancreas also secrete pancreatic amylase. And the main function of pancreatic amylase is to convert starch. I'm going to write down the amylase. The difference is that is, as it is secreted by this gland, so we are using the term pancreatic amylase. LASE, not very clear, right? Amylase, right? So this pancreatic amylase converts starch into disaccharides. Disaccharides. So there are so many different types of disaccharides, peptones and proteases. Too much of the amount of the fat is also digested over here into the duodenum, and then the food reaches into the ileum. So now I am going to start digestion and with different enzymes which is there into the ileum. So ileum contains, I am going to write down the last part of the small intestine. That is ileum. Now, the ileum is actually going to remove the upper part of this digestive system, so we'll be getting the space for writing, right? Ileum. Now, going to tell you the different enzymes. First of all, if the bit amount of the fat is remaining, so again that is fat will be converted into fatty acid and glycerol. The same way, this is fatty acid plus glycerol. And the name of the enzyme is again a lipase. It will be remain same. This is the important one. Complete digestion of the fat is there. The organ is ileum. Now we have so many different types of disaccharides which are actually formed after the digestion of the starch. The names are number one work is to be done over here. Second one, that is termed as maltose. Maltose is a type of disaccharide. For digestion of this maltose, the wall of the ileum secretes one enzyme and the name of that enzyme is maltase. This maltose is converted into two molecules of glucose. That means glucose is monosaccharide. When two monosaccharide molecules, glucose plus glucose, are united or definitely the condensed, then one molecule of maltose is to be formed. So definitely when we digest maltose, the two molecules of glucose will be formed. The name of the enzyme is maltase. The second disaccharide digestive enzyme is, the disaccharide is sucrose, cane sugar. It is sucrose. Name of the enzyme is just resemble with this. Only thing is to add the ase. So the term is sucrase. This is there, and as it is disaccharide, so definitely one molecule of glucose plus one molecule of fructose, that is fruit sugar, Fructose. This one is monosaccharide, this one is monosaccharide, and this one is again monosaccharide. Third type of disaccharide, that is termed as lactose. Lactose is milk sugar. Name of the enzyme is lactase. You can identify this. Lact is taken from here. Ase word is indicating you the enzyme. So this is lactose and this is lactase. This one is enzyme and this one is disaccharide of milk. This lactose is converted into again one molecule of glucose and one molecule of galactose. Galactose is also a milk sugar, but it is monosaccharide. So the complete digestion of the polysaccharide is over here. Starch, then starch to disaccharide, then converting to the monosaccharide. The complete digestion of the fat is also here. The fat ultimately, the whole soul fat what we have eaten is converting to the fatty acid and glycerol. And if the amount of the protein which is there, going to remove this last line again, the protein which is left, it is there in the form of the dipeptide. This is dipeptides, and of course maybe the polypeptides, right? Converted into amino acid. Amino acid. And if little bit protein is also there, rest of the protein, that will be also converted into amino acid. Maybe the peptones or proteases are also left. That will be also converted to the amino acid. And the name of the enzyme is eruption. So this is the whole soul digestion is over. So ingestion and then we completed digestion. Ultimately, you know that in small intestine, a villus-like structure is there. When the food is completely digested, then this villus is responsible for absorbing the food into the blood vessel. That means if it is a digested food, which is ultimately into the small intestine termed as the chyme, then it will be digested itself across this microvillus into this blood vessel. Fatty acid and glycerol are not absorbed into the blood vessel. For the absorption of the fatty acid and glycerol, we have a lacteal because it is not soluble into water. So we have lacteal and it is ultimately absorbed into that one. Thank you and bye-bye.